my pleasure to introduce a friend for today's seminar, Brian Pfleger. Brian's a rising star in chemical engineering, particularly in our sub-area of interest, bioenergy. And not surprisingly, he's won several awards. Now, I'm not going to list all of them, but amongst them, the NSF Career, the DOE Early Career, and recently the Daniel I.C. Wang Award. But perhaps the one that's most treasured by Brian is that he was awarded tenure a couple of years ago from University of Wisconsin. Of course, we're all very proud of Brian. He's an alumnus of Berkeley. He got his PhD in 2005 from Jay Keesling, and then went off to do a 2-year postdoc in David Sherman's lab at Michigan, and then has since been at Wisconsin. So please help me welcome Brian.
Thanks, John. It's great to be back in Berkeley. It's really impressive to see the new additions, both the physical and the personal, to the campus. It's always has been an exciting place and something that will be near and dear to my heart. So we've had a great day so far, and I'm looking forward to the conversations this afternoon. And so the things I want to talk about today, usually I give about a 15-minute introduction to my field. I'm going to try to blast through as much of that as I can because I think most of you are very cognizant of the area and the motivations for the work that we do. But I am going to talk about some of the issues because I think it's interesting to see different points of view. And so for the students and researchers, collect these thoughts and take them with a grain of sand. And then after that, I'm going to talk a little bit about a couple different vignettes, touching on things from metabolic engineering to synthetic biology tool development, to some work with catalytic scientists that we've been doing to look at different ways of processing biomass. And so to get started, I have to acknowledge the people who did all the work. I love laboratory work. I love experiments, but unfortunately for me, I haven't been in the lab in a very, very long time. And so these are the folks who have done everything you're going to hear about today. We're a mixed group. As you can imagine, energy or metabolic engineering topics are interdisciplinary, naturally. And so all the interesting problems, in my opinion, at this point in science are at the boundary of different disciplines. And so we have chemical engineers, biochemists, chemists, microbiologists, and people from all sorts of different backgrounds in the lab. And so a project in the lab starts with designing a particular objective. Sometimes that's a biochemical objective, sometimes that's a metabolic engineering objective, but all the times we need some sort of genetic component to add to our system. We're working with in vivo systems. And so that means we sometimes have to discover genes and characterize them so that they'll function in our particular system of interest. We then will have to oftentimes develop new tools to figure out why they aren't working, right? Most of you have done this. You put a gene into an organism and it doesn't work the way you expect. So what is it about the gene expression, about the protein's function, its solubility, and how do you develop tools that help engineers do this faster? We've applied a lot of our work in metabolic engineering towards the production of oleochemicals. So these would be things that would be either in the biodiesel or fatty alcohol class, and I'll talk a little bit about some of the success that we've had there. Most of the times when we design a new strain for producing a particular oleochemical, it stinks. It doesn't come anywhere close to the objectives that we were setting out for ourselves. And so the question becomes, how do you dissect where your barrier is? What's the fundamental bottleneck that's preventing the cell from meeting those objectives? And so we'll use systems biology or functional genomics approaches oftentimes to kind of search for that needle in a haystack. I won't have as much time to talk about that, but I'm happy to, over lunch, talk a little bit about some of the things we've done there. And then we try to interact with real process design, so trying to take things that we do in a shake flask and put them into bioreactors and work with more process synthesis folks at Madison. And so I wanted to start because the title said sustainability through synthetic biology. And so if you take a panel of 10 self-prescribed or self-defined synthetic biologists, you'll get 9 different definitions for that field. And so I want to give you my perspective on this field, and that is that it's just the most recent evolution of what we would call bioengineering. And so this all goes back to recombinant DNA technology. And so actually a UW biochemist, Václav Sobalsky, wrote in a textbook right after the first transgenic experiments were performed that this synthetic biology era was about to begin. And he was putting that in an analogy to the way that synthetic chemistry redefined the way that chemists looked at systems. Instead of just exploring the natural world, they were starting to put together new reactions and new chemistries. And so this was going to be the era for new biology. And so this, of course, led to the current biotechnology industry, where most products were the product of one gene. So you put one gene into a transgenic system, and you made a protein, or you gave it a particular function. And so in my opinion, synthetic biology is just now the next step, the evolution. We're not dealing with one or single gene type technologies. We're dealing with many gene technologies. And so why is this something that people perceive as new? It's now the inclusion, in my opinion, of new disciplines in this area. So folks who are chemists, folks who were biotechnologists, folks who were microbiologists, this is not anything new. It's just kind of the next stage in their thinking. But to someone like an electrical engineer who's just seeing this for the first time and seeing the parallels between the natural world and the synthetic circuits that they were building in the electronics industry, this is an amazing new playground. And so it's to these folks and their inclusion of their thinking, the parts-based thinking, that makes this field so exciting and new to them. And so I think most of you are familiar with this work. This is the project that I happen to be part of, or very loosely affiliated with in Jay Kiesling's lab as a student. There are lots of ways that you could apply synthetic biology, and the focus of what he was doing at the time was trying to take biochemistry that was in a difficult system, so this plant-based system, and trying to transfer the genetic basis for that biochemistry into microorganisms, so that then we could take the same types of technologies that we had established for producing fermented beverages and instead produce this molecule that would be wonderful for curing diseases. And so this type of approach, where you're taking not just a single gene, you're taking an entire pathway, and oftentimes genes that we don't even know, we have to discover them, optimize them, and put them all together and optimize their function inside a living organism, was some of the first applications of metabolic engineering. And so lots of folks have thought about this in terms of natural products. Can we discover, the next penicillin, things of that nature, or do combinatorial biosynthesis for that type of natural product discovery? But there's also a lot of success that's happened in industry for much simpler, much smaller molecules. And so the other model story is that of 1,3-propanediol from DuPont. And so this is work that actually was started at Wisconsin by Doug Cameron's group in the labs that my students now sit in. And so it's near and dear to our heart for many reasons. But the reason that this process is such an industrial success is because it hit three major objectives. It's a molecule that is functionally better or superior than its counterparts. It's a molecule that's now made cheaper, so the economics are better, and it's environmentally friendly. So it's displacing petroleum as a source material and instead using a renewable or sustainable approach. And it's because the combination of all three of these come together in one product that you've seen such an economic success. Most products that you're going to look at, the simple, easy, low-hanging fruit, things like biofuels, they don't necessarily meet all three of these objectives. And so that's one of the challenges we face as we try to pick out the next and the best targets. And so if it's my opinion that synthetic biology is not new, I will grant it seems to be emerging or exploding in the common lexicon. So why is that? Why is it such an exciting time to be in this type of bioengineering? And so I'll give you three different technologies or things that have happened in the last decade. First and foremost is we can sequence just about any organism out there. So if you happen to be hiking in an exotic location and you discover some amazing natural biology, you can take a scoop of that dirt illegally, bring it back to the United States and sequence that organism and potentially leverage the biochemistry that was in that organism. And so from all of this sequencing technology now begets RNA sequencing and all of the more functional genomics-based technologies that aren't just looking at microarray comparisons. We're actually looking at sequence differences at the base pair level. And so it's a really exciting time in what we can do in terms of DNA sequencing. All of that sequence information has to be put someplace. And so now we have this collection of different databases. And so whether you want to search for something based on sequence information, functional information, sometimes even structural information, there are databases that are being put together for all of these things. And so in the past, where you might have only had one example in the literature of a particular enzyme, you can now go to these databases and using different types of BLAST algorithms or search algorithms like that, find entire families that are maybe evolutionarily similar, or maybe they're very distinct, and you can choose which ones you want to then play with in your system. You don't have to limit yourself to one particular source or one particular gene. Furthermore, in the past, when I started as a graduate student, we had to either go isolate the original genetic material that encoded that gene, or pray that one of our collaborators or competitors would be willing to send us the constructs that they had cloned themselves. That era is now over because you can go to any DNA synthesis company and synthesize any piece of DNA, now oftentimes with the variants that are maybe active site variants or inactive mutants, all sorts of different variants on the theme for next to pennies, right? So the cost of synthesis and the scale of synthesis, not just simple oligos, but now it's getting into genes and even chromosomes, and soon certainly whole organisms, genomes. And so it's these three technologies that have enabled, and I'm sure I'm missing a lot of different specific examples here, the development of metabolic models, the development of different types of functional genomics assays for interpreting how cells are functioning. It certainly enables the combinatorial biosynthesis, so if you want to do pathway optimization, you can now synthesize 3 different genes for a particular enzymatic step in a pathway, and then explore through a library which one ended up being the best. And so there's lots of different applications here. And so the real question becomes, if this is such an exciting, hot area, where can we apply this type of technology? And so the area that I've been most interested in, since I'm a chemical engineer, is trying to replace the petroleum industry, or as much of it as we can, with things that are going to be more sustainable. And so our idea here is to come up with green chemical factories. And so this isn't the only area, of course. This is a slide from the grand challenges from the NAE. I always like this slide because it conveniently left a blank space for me to put in my own favorite challenge. And so I say that this is equal to many of the others, but certainly not inferior to things that we're going to face in the future. And so in chemical engineering, when you think about energy and the petrochemical systems that have been developed, there's probably 2 things that you need to remember that are often overlooked. One, the absolute enormous scale that these processes run on, and how incredibly efficient we've made them. And so the reason we have cheap energy, cheap gasoline, cheap diesel fuels, is because a fraction of that barrel of oil that comes into the first petrochemical refinery makes its way into the commodity chemicals that then get produced into all sorts of higher value chemicals. Right? And so it's that economic revenue that's generated from a very small slice of the distillate that helps afford the low cost of all of the other fuels that are being produced. And so when we try to recreate this or replace this, you have a hard time because you've got to try to create things that are both high volume, but also very high value. And so the reasons that we want to replace that very efficient process are varied, and I'm sure in this room you can argue about which one of these is the most important. And so whether it's world supply, so when I started my faculty career, we were right here, and we were wondering if we were in another peak oil era about to start seeing the first declines. Whether it's energy security, right? Of course, the United States, sorry, the laser there, is a net importer, but that trend has been changing in recent times. And so lots of new petroleum discoveries are changing the landscape of where petrochemicals come from and are processed. The economic arguments are still very much in play, and of course the gorilla in the room is what we're doing to the planet in terms of releasing carbon dioxide every year. And so whatever your motivation is, I think the idea that we want to come to is a way to take carbon, use it, and then reuse it in different cycles. And so if we're going to get energy out of this cycle, that means we have to put energy in at some place. And so that has to come from photosynthesis or from solar energy in some form until something science fiction-like comes around. And so the idea here is that there's lots of different technologies, whether you're using terrestrial plants, aquatic species, whether you then take the biomass that's in these molecules or you make a chemical directly linked to photosynthesis. There are lots of competing and collaborative technologies that are going to get us to a mixture of these crude biofuels and chemicals. We're going to refine them, use them, combust them, get the CO2 back out that we can then use with the sun to repeat the cycle. And so this has been the goal for biofuels since its inception. And sometimes the students that I talk to lose sight of the fact that this is one idea inside a much larger, much more complicated system. And so you don't just have the technical advantages to argue here, you also have to look at things like economics, like legal implications. And so just looking at what other things could lead to crude biofuels and chemicals, we could discover new petroleum reserves that would then drop the petroleum prices, and that's we've seen some of that, and so that economic argument comes into play. More important, we have a large system for producing natural gas at incredibly low cost. So this is probably going to be the economic floor for any sort of chemical production. How expensive is it to make it from a Fischer-Tropsch synthesis or something like that? And of course that would also play into then other types of methane generation or natural gas generation. There's the whole idea of using solar energy more directly, and so using electricity in various ways to reduce carbon dioxide. and of course, efforts to then recycle, reuse materials instead of combusting them all the way back to CO2 and requiring that energy. So there's a lot of things competing here, and so to say that biofuels or bioenergy is going to be the silver bullet is a complete mistake. I think we have to be cognizant that all of these different technologies are going to play a large role in what we do. And the other sort of take-home point before I get into more of the specifics is that we don't have to produce only fuels. There's a wide range of chemicals out there, and there's a wide range of technologies. And so what we need to do is think about matching the benefits of particular technologies to the molecules that would be the best examples. And so these are all molecules that could be made biologically. I don't think it makes any sense to have an organism convert something like glucose or even carbon dioxide into methane. We have plenty of that in various places. It's pretty obvious that Taxol and complicated proteins like antibodies are going to be the domain of biological synthesis. I think that's fairly well established. But there are lots of simple molecules in the middle that we really haven't established which is going to be the winner. Ethanol, which is a very simple molecule and can be made from ethylene, for the most part is made by fermentation because that happens to be the more economic route. The same for 1,3-PDO. And so folks are starting to ask questions about similar molecules like styrene or acrylic acid that have now been introduced in combination processes of catalytic science and biological science. And so I think what we have to start thinking about is how do we train people to be able to see these intersections and take advantage of the things that chemical catalysis can do really well. So high concentration, high speed, high scale, those are all the powers of heterogeneous catalysis. But they can't make bifunctional molecules all that well, right, without the protection, deprotection strategies. And so to get at modest conditions and to make very complicated molecules, enzymes are going to hold a lot of different advantages. And so the question becomes, where on this chemical spectrum, how do we design an entire chemical industry that leverages all of these? And I think it's going to be one of these things that evolves over time, of course, and so we're all trying to find that low-hanging fruit, that thing that's going to demonstrate our ideas or our technologies. But in isolation, that might work wonderfully. But if all of a sudden we reduce the price of this particular compound by making it biologically, it's also going to drop in price what you could sell it for. And so the economic arguments that are obviously things that need to be worked out. All right, so I'm going to transition now into some of the things that we've been focusing on. And so what's really interesting in this space called oleochemicals is that these are molecules that organisms have made for a long, long time. But the biochemistry and the genetic basis for how they're made has really only been uncovered in most of these cases in the last, say, decade, 15 years or so. And so there's been an explosion in metabolic engineering targeting various molecules like methyl ketones, different types of ketones and olefins, alpha olefins, fatty alcohols, alkanes, other biodiesel-like molecules, and even structural types of materials that are made from fatty acids. And so we got interested in this because we were looking for a platform, so something that we could make, and so we decided to look at free fatty acids and then leverage our strains that would make free fatty acids into producing all of these other types of compounds. And so the story I'm going to tell you is about fatty alcohols, and so the reason I'm going to talk about fatty alcohols is they're the only one that makes economic sense in today's market. And so what you see are on these three graphs are fatty acids, biodiesel molecules, and fatty alcohols of different chain lengths. The prices for those were acquired from a consumer market group. And so what I've plotted here is using a genome scale metabolic model, the theoretical yield of each of these compounds off of glucose. And then converted from the sugar price here that's on the x-axis, how much money would have to go into producing that compound, and then how much is left over after you sell it. And so, of course, if you had a zero glucose price, you would have 100% of your selling price left over for selling it. And so if you look at something like 15 to 20 cents per pound glucose, which is probably realistic, these are all the different curves for fatty acid, ethyl esters, methyl esters, acetate, alcohol esters, alkanes, and methyl ketones. The slopes are different because the theoretical yields of each of these compounds are different, but you'll note that they're all roughly the same. And so if you look at somewhere in this 15 to 20 cent range, you're looking at prices where you only have about 20% of your selling price left over. Now, this is just raw materials, and not necessarily all the raw materials. We haven't factored in energy or other operating costs. And so if you compare it to a petrochemical refinery, right, where the raw materials cost roughly 60% of the final selling price, we still got a long ways to go. And by the way, these are all at theoretical yield. Most of the best processes are somewhere between 50 and maybe 60, 70% of theoretical yield. So we have a long way to go. Now, if you instead look at molecules like fatty acids, they're also very low price, low price selling points, and so they're usually blends. And so you'll note that most of these are long-chain, very common fatty acids. There aren't a lot of actual price data points out there for these smaller fatty acids because they're not as widely sold. But the fatty alcohols are, and so if you look at things like decanol or octanol, you'll see that the slopes of these are much more shallow, and that's because the price points for these are 2 and 3 times higher than what you typically see for the long-chain fatty acids. And so if you look at that same price point, right, 15 to 20 cents a pound for glucose, you're starting to talk about margins that are similar to the conventional petrodiesel. So this is why companies are interested in these, perhaps as the demonstration process that then they can hopefully show a plant that works, change out a couple of enzymes, and instead of making a fatty alcohol, produce some sort of biodiesel molecule. So how do we make fatty alcohols if we're going to be interested in making these? So there's a series of fatty acid biosynthesis is an elongation process, so it's cyclic. You basically take acetyl-CoA and you polymerize it through an activation with a carbon dioxide to make malonyl-CoA, and those malonyl-CoAs are then the extension unit. And eventually you reach a long chain where those intermediates are then fed into lipid synthesis. And so for fatty alcohols, what you end up doing is you hijack those same substrates that are used to make lipids, but instead reduce them to make aldehydes, and then a second reduction to make fatty alcohols. Alternatively, you can feed or find fatty acids in the environment, activate them as acyl-CoAs, and there are acyl-CoA reductases that would do the same types of chemistry as the acyl-ACP reductases over here, making fatty aldehydes that then can be converted to fatty alcohols. And of course, Mother Nature would explore this space completely. There are direct proteins that do both chemistries, so making an aldehyde internally to the enzyme to produce fatty alcohols directly, and you can skip this separate activation step here and make fatty aldehydes directly. And so basically the stoichiometry is all the same. If you're going to start with a free fatty acid, you need 1 ATP and 2 reducing equivalents, and you can make yourself a fatty alcohol. And so if you take some of the best studied of these enzymes, and so this is our favorite, this one that does the CoA conversion directly to the fatty alcohol, you'll note that it also has activity against the native long-chain acyl-ACPs. And so if we feed a mixture or a bolus of lauric acid here as free fatty acid, we can produce next to no fatty alcohol in the wild-type cell, and that's because this beta oxidation process will consume it faster than it would be converted. But it also will produce a little bit of the long-chain hexadecanol coming from the native long-chain acyl-ACPs. If we remove that beta oxidation step, but not remove the important activation here, we can then flip that and make some fatty alcohol, but you'll note that if we feed a lot of fatty acid, it doesn't get consumed, and very little of it ends up in our desired product. Yet in the same strains, we still produce about the same amount of hexadecanol. If we, however, then alter the expression of this particular enzyme here, the activation step for the free fatty acid, we can end up switching that and producing almost entirely fatty alcohols from the material that we fed. And so the point I want to make on this next slide is, so we've taken one enzyme that was already there in the natural cell, and we've shown that we've had to upregulate it in order to get some sort of beneficial effect. Well, how much do you upregulate these enzymes? A lot of folks will go in and they'll use a T7 system in E. coli, and they'll make an enormous amount of protein for a particular reaction. And oftentimes that's very, very bad for different systems. So here is just a titration experiment where we put the wild type as the normalization agent. We deleted the gene, and of course we don't get any fatty alcohol produced. We just replace the chromosomal promoter, and so in doing so we get about a 50-fold enhancement in transcription. And here where we put it on a plasmid, where it's 1,000-fold relative to the wild type, you can see that the best scenario for producing fatty alcohols instead of fatty acids is at this sort of intermediate point. And this is a trend that's been seen oftentimes in the literature, that the optimal enzyme expression is not maximum activity or maximum protein levels, it's some sort of intermediate. And so that's what makes the optimization step so challenging, is we don't know what level needs to be there ahead of time. And so it becomes dependent on different types of either screening or rational approaches to come up with ideas or hypotheses for what the optimum would be. So all the work I showed you here was with fatty acids being fed. Of course, that's not really useful. We want to convert glucose into these molecules. And so some of our early work exploited the literature using a thioesterase from the California bay tree. Happens to be a very, very active enzyme, very happy in E. coli once you remove its membrane tag at the N-terminus. And so if you take that particular enzyme, put it into a FAD-D deleted strain, you can produce lots of fatty acids. And if you overexpress other rate-limiting steps, you can enhance that process even further. Even better, these fatty acids are excreted outside the cell. And we showed that with a combined catalytic process, if we wanted to make a biofuel out of these fatty acids, we could extract them with a layer of undecane, then pass that over a simple catalyst that would do the decarboxylation ex vivo, separate some of that alkane back off as the next round solvent, and send the rest out for product. And so we don't necessarily have to do all of these conversions in vivo. We can leverage combined processes and be very effective. And so when we take this strategy, we obviously can't delete FAD-D, so we switch it and delete FAD-E, the next step that would send the acyl-CoAs back around beta oxidation. We can start to produce fatty alcohols directly in vivo. And so we did this by putting different numbers of thioesterases in different loci in the chromosome. So we were trying to reduce the number of plasmids that we had to have in this particular system. And so when we do a fed batch, that's the data that's down here. We just added some simple boluses of glucose every few hours, so it's not a very elegant fed batch. We were able to get linear productivity of over 100 hours at roughly 40 to 50% of the theoretical yield of the fatty acids. There's still a lot of room to go. We are producing a lot of acetate, and so that's a simple deletion that we can make. We're currently working on those types of things. But the other thing that was interesting was a process note. And so this chart down here, sorry, the pointer's not working all that well, is another feature that seems to be common in a lot of these the production of hydrophobic molecules. So the light-colored bars are a strain that's producing fatty alcohols in just a normal fermentation broth. The dark blue bars are the same strain in the same media, but with a 10% dodecane overlay. So we're adding a little bit of a solvent here. And so you'll note that the y-axis is the percent in the supernatant. So this is the fatty alcohols that is not cell-associated. So if we take a sample, we pellet the cells, and then we look at what's in the media and the dodecane versus what's in the pellet. And so you'll see that in the case of just the strain, that we're about 40% of the material is excreted. Yet if we provide this solvent, we also see that it's now almost entirely in the extracellular space, so helping our purification. What's more is if you compare the titers, these last two bars, the last one is the addition of the dodecane, we also see a dramatic yield increase. And so what's happening there likely is that you're either removing the fatty alcohol is the last step from aldehyde to alcohol is reversible, and so you're either pulling the biochemistry in the one direction, or you're pulling the fatty alcohols out of the cell and improving the cell's fitness. And so those are the current hypotheses. And so there's still a long, long way to go. As I said, this is roughly at 50% of the theoretical yield for these particular processes, and so that's not going to make us profitable anytime soon. And so the approaches that we're looking at now to improve theoretical yield are trying to figure out where's the carbon going and why is it going into those destinations, and can we make genetic manipulations to direct all of the carbon in the areas that we want it to go, and can we then design bioprocesses that would maximize the ability of each biocatalyst. And so that's the genetic engineering side. There's a second knob that we have access to, and so I've flipped this around. This is, of course, just for biofuels. The price, so this is the contribution of the raw materials, the glucose, at theoretical yield. And so the other challenge, of course, is driving the cost of sugars to the left. We need to decrease those sugar costs. And so one of the things that I noted, and I think all of you are conscious of this, is that not all biomass is created equally. And so if you're going to go after cellulosic biomass, there's a lot of processing differences that have to go into getting that cell wall broken down and accessing those polymers. And the enzymatic routes for doing so are very, very good, but not necessarily feedstock agnostic, right? So if you have grasses today or corn stover tomorrow and trees for the next day in your plant, you're going to have to optimize the cocktail, the conditions, et cetera. And so on top of that, the costs of the enzymes, this is work that Harvey Blanch did, it blew me away how much out of proportion these estimates were when he had looked at producing enzymes versus what the common literature was. And so in my mind, I started thinking about how can we get to a cellulosic process that doesn't involve these enzymes. And so conveniently, I have a brilliant collaborator, Jim Domestic, who is a pioneer in lots of areas of catalytic science. But this is one of his latest and greatest. And so this is a molecule called gamma-valerolactone. And so this molecule, most importantly, can be made from sugar. So you can convert glucose to this in very high yields. And so what he discovered, or his students discovered, is that if you stuff a small pipe, that's just a steel pipe, with any type of biomass, doesn't matter if it's corn, stover, grasses, hardwood, softwoods, etc., feedstock agnostic, and you start to flow this material in through this tube, and you start to heat it up. So this is not a batch process. It's kind of a semi-continuous process at the moment. What will start to come out at the bottom are different fractions in the solvent containing different amounts of the biomass material. And so the first thing that comes off that I don't show you here is the lignin fraction. And what's more impressive about this right now is that the subsequent analysis to when we published this paper, the lignin looks identical to the best purification protocols for lignin that are out there. The NMR spectra are literally superimposable. And so it looks like a really exciting technique because most of the lignin comes out within the first few volumes out here, and that's why it's not shown. And so as you ramp up the temperature and you add acid, so note that this is 2 orders of magnitude less acid than you would need for comparable conditions without the GVL. You ramp the temperature up and you get some breakdown of the hemicellulose, and so you start seeing pentose and pentose oligomers. And then as you continue to heat the system up, you start to see a broader distribution then of the hexoses and the hexose sugars coming off. And so even better than all of this is that the GVL solubilizes everything. And so one of the kind of secret challenges is how do you deal with a 20% biomass slurry, right? That's not something it's oatmeal. It's not something you're going to pump. So these are ways that we think we can, on farm perhaps even, solubilize a lot of the material and send it off in a way that could be pumped. And so to compare this, if you just take water and you run the same type of temperature ramp with acid, you will produce the sugars, but you're also going to produce a lot of gunk, right? And so this gunk is humans and other sorts of breakdown products. What the GVL seems to be doing is enhancing the breakdown catalysis so that we don't have to use the harsh conditions that end up leading to these materials. So we can end up breaking down the biopolymers into their fermentable substrates. So it's not all glucose and pentoses, but we recover close to 90% of all of these sugars. Now that leads to the next challenge. Jim's group is going to take this solvent sugar mixture and either make more solvent or convert those into butenes that he can then oligomerize and make all sorts of diesel and jet fuels. And so that looks like a really exciting, attractive process. What happens if we want to use those sugars in a biotech process? They're stuck in this GVL solvent. So the organisms, unfortunately, don't like that. They're not going to grow in a large concentration of GVL. And so we needed to come up with ways to salt out or precipitate the sugars, transfer them into an aqueous phase. And so, of course, the first thing we tried was sodium chloride. This worked really well. You can separate almost all of the sugars into an aqueous phase and get relatively high concentrations, but you require ocean or 2x ocean salt concentrations of sodium chloride. So clearly that's not going to work because E. coli is more or less dead at 1 ocean concentration. And so the next step was to try some other solute that would be innocuous. And so here we tried supercritical CO2. And so it worked wonderfully. And we have advanced the process now so that we can get over 100 grams per liter of carbohydrate concentrations and use something then that doesn't have to be diluted to deal with either salt or other impurities. And so what's amazing about these is they come out relatively clean. They're still colored, so there are some toxins, and we're dealing with some of that. Most of the actual toxicity that we associate with this paper, for those who read this, turned out to come from the metals. And so we have some recent experiments where when we switch from a steel tube, which the GVL will leach some of the metals from, to a more innocuous metal, that we end up with no toxicity. We don't have to dilute these materials at all. And so this is where we're going. We're really excited about it because, of course, we can ferment these with ease to ethanol at reasonably high yields, and the techno-economic analysis put us right in the ballpark from the get-go. So the challenges, of course, now are dealing with energy recovery. What are we going to do with the supercritical CO2 and managing that? The estimates we have used for this model are, there are only a couple of points around a supercritical point, and that's not the best most accurate situation. And so there's a lot of work still to be done, but we're really excited about this particular process. And of course, we can make fatty acids. And so transitioning now to the next side of the story, we don't have all the biomass in the world to make all of the energy molecules or all the petrochemicals that we want. And so I think we need to start thinking about what are we going to use terrestrial biomass for, and can we create new sources of terrestrial biomass that would even enhance these estimates. And so my group's been thinking a lot about how do we skip the biomass middleman. And so there are lots of inherent challenges here, but the general idea is to take waste nutrients from other streams that we want to clean up, carbon dioxide that could be available from various power plants and sunlight, and make all sorts of different things, including cleaning up water. And so the other way of framing this question is, we've been thinking about a system where we take photosynthesis and we make some sort of low-value, high-volume intermediate. And then we have a separate plant that processes all of that material into the things that we want. Could you not instead have a small-scale facility that would do the same photosynthesis, but instead of making the low-value intermediates, you would produce your desired chemicals directly? And so this is the idea for photosynthetic biorefineries, where we would have some sort of either open raceway pond or some sort of closed photobioreactor systems. And I think the challenge here is obvious. These are going to be incredibly more expensive than your traditional fermenters, or even more so than the traditional fermenter plus the agricultural costs associated with harvesting. And so how do we reduce these costs or deal with the contamination and other issues that are associated with raceway ponds? And so we have a project with several different folks at Madison, a systems engineer, a wastewater engineer, and a photocatalytic reactor designer. And so we're trying to address a lot of these. But to start to look at those, we need real data. And so we needed to start to engineer cells to produce chemicals in any form so that we would have realistic numbers to work with. And so I'm going to skip this for the time being. The strain that we decided to work with is a model cyanobacterium called Synechococcus PCC7002. Ignore cyanobacterial phylogeny. The Pasteur number is probably the easiest way to know that you've got a different strain from one another. And so the reason we picked this number, and unfortunately this looks like it all shifted, is it's one of the fastest growing cyanodes out there. There are some folks who have isolated cells recently that are a little bit faster than this, but up to this point, it's the fastest grower. And so you can naturally transform these guys. They can use either wastewater, salt water, or fresh water. They can grow in any of these medias, and they have a very, very high light tolerance. And so that's important for dealing with the laboratory conditions, so we can get these things growing really, really fast. The problem with these, of course, was we didn't have a lot of genetic tools. So we didn't have a synthetic biology toolbox that consisted of promoters or selection agents. And so we had to start working out a lot of these. And so the first challenge that we encountered was we could get a gene into the organism very easily, but there are 10 copies of the chromosome for these cyanobacteria because they have to deal with high light. And so the challenge becomes, how do you segregate and get a homozygous set of strains? And so to do that, you really like to have some sort of counter-selection system. And so we were looking at producing acrylic acid in these organisms, and we happened to just test the tolerance of these and discovered that there was a mutant that would pop up every once in a while on plates with a certain concentration of acrylic acid. And so when we took this mutant back into the conditions and looked at its inhibition concentration, it was an order or several orders of magnitude higher than the wild-type species. And so we mapped this mutation to an acetate-CoA ligase that has activity against acrylic acid. And so we think that either you're producing a dead-end product that could be inhibitory to other enzymes, or you're just depleting the free CoA pool inside these cells. We think that's the issue. The mutation happens to be someplace that we can't understand, so it's probably something to do with the solubility of the protein. But what we can do with this now is have a natural chromosomal location to drop in any gene of interest, and all we have to do is select on acrylic acid, and we can segregate mutants really, really quickly. We've done this then by inserting either spec resistance or other resistance markers with this locus into other locations, and so we can delete those genes very rapidly. So we now have a system where we can make chromosomal mutations on the order of a week instead of, it used to take us at least a month to get a different integrant constructed. And so we use this system then to explore synthetic biology strategies for producing different gene expression control elements. And so the first easy thing to do if you want to know what promoter to use is to have a library of constitutive promoters. And so we took the Anderson library that was created here, as well as doing an error-prone PCR approach, and looked at different families from those two sets. Of course, when we took those same families and rank ordered them in 7002 and put them in E. coli and rank ordered them there is absolutely zero crossover. So unfortunately, we can never just take knowledge from one organism and transplant it directly into the other. But we did have a nice family that spanned several logs, and so what we could do with these then is take this system where we took a strong promoter, this happens to be a photosystem promoter, that we engineered to remove its light sensitivity, and we placed next to it different operator sequences that would respond to LAC-I. And so when you add these operators, of course, we break the promoter severely, and then when we add LAC-I, we can repress it some more. We add IPTG to these cells, and we can restore expression. So this is a nice 3 to 5-fold induction. And so what we did from there was adjusted the promoters using the libraries we created for LAC-I, made some point mutants in LAC-I that have been shown to make its binding affinity even stronger, adjusted some other things in the original promoter, and made some small additional point mutations to the point where we now have 2 that we're really happy with that span almost 40-fold of an induction range. So we can use these for different purposes. We did something similar recently with an undergrad in the lab to adjust the TET-R system. So now we have an anhydrous tetracycline system that can be used in parallel with these LAC promoters. And so what we've done with these then is to try to work with lactate production. And so we picked lactate because the downstream processing and a lot of the work there has been industrialized, and so there's good case studies to look at. And so we know that lactate may not be economically the right choice, but it provides us with a molecule that's fairly easy to get to. It's only a couple of enzymes that you need to add to 7002 to make lactate. And so we did that, and we now have the best lactate-producing cell that's out there. Unfortunately, you'll note that it makes 262 milligrams per liter per day. And so that's not going to be economic anytime soon. So this is the challenge. Most of the cyanobacterial production strategies are on the order of gram per liter per day. And so that is going to be our limitation until we figure out ways to enhance carbon fixation, which we've been trying to do for a very, very long time, or we find ways to justify and make really low-cost photobioreactors so that the economics of producing all year instead of just harvesting 1 or 2 times a year balance out. So these are the things that we're thinking about and we're working with in the lab. And so I'm going to close with one last vignette, and this is one that I'm really excited about. And so I'm super excited to talk to Jennifer Doudna later this afternoon. I've never actually had the chance to meet her, and so the things we're interested in here would encompass her CRISPR-Cas work in some respects. This is the idea that we have lots of genes that we artificially put on plasmids, or we mutate the promoters to get particular expression profiles that we want. There are other situations where that's not that easy. So you want to go in and you want to mutate something on the chromosome, and you don't want to remove its native regulation. So maybe there's something about relating growth rate to expression that you want during the growth phase, but you want the thing to be off in the production phase. And so how do you deal with that? How do you control regulation, control gene expression, but do it in trans, not with mutating something in cis? And so we've been interested in this, and there are lots of different technologies for this. You can, of course, use small RNAs. There's the CRISPR-Cas system. But what I'm going to focus in on are these proteins called TALs. And so the idea here for a TAL is it's analogous to a gene finger. It's a protein that has the ability to bind any DNA sequence, except it works. And so that's what's exciting about it. So these are natural proteins. They're made by plant pathogens, and so they typically get secreted into the plant tissue. They make their way to the nucleus. They bind to a particular loci in the genome, and they have an activation domain flapping around right off of that binding area that then recruits RNA polymerase and turns on expression of genes that then typically lead to the susceptibility of the plant to infection. And so this is the way that pathogens are trying to influence their environment. And so what's exciting about them, and what makes them different from all of the synthetic biology strategies that have used DNA binding proteins that are very well established, is that these are restricted to these DNA sequences. Now a tau is something that we can then engineer to bind to any particular sequence that we want. And so these proteins are incredible. These crystal structures are spectacular. It's a multimeric protein, so it's basically the same 34 amino acids with the exception of these central 2 repeated in each of these colors. And so you see somewhere between 15 and 25 of these repeats. Each repeat projects 2 residues into the major groove of DNA, and there's these contacts that then are on a nucleotide and amino acid residue code that's analogous to a Watson-Crick code for binding a particular base. And so these things spiral around the DNA and provide a wonderful ability to bind to particular pieces of DNA. And so what folks have done in the last few years and why they've gotten excited about them is that the TALs they came on board a little bit before the CRISPR-Cas phenomenon, so they had the nucleases attached to them instead of activation domains, and so they were the TAL-Ns that you might know of from mass market emails. And so the TAL effectors really weren't explored then much for their natural ability, which was gene expression. And so I asked the student to just, we had a group meeting about this, and somebody presented a paper about TALs, and we were excited about it. Well, I wonder if it would work in E. coli. Why don't you go design one to the LAC operator and just see if it works? And so he did that, and he designed a particular TAL. We had to have the gene synthesized, of course, as these are fairly large proteins, and it's a very repeat-rich sequence. And so we had it made, and he expressed it from one plasmid and had a LAC-I control, so it's basically the same plasmid but with LAC-I present. And he designed a reporter vector that had either mCherry or GFP and the particular LAC operator from the TRC promoter, which is known to be a very strong promoter. And so you put these two things together, and of course, when you express the TAL, you get next to no expression from that GFP. And if you compare that then, this is a time series, each of the colored bars representing further in the growth cycle. But LAC-I has a lot of leaky expression as you get into later times. And so we found that this actually is enhanced in terms of its ability to repress DNA. Of course, this comes with the caveat that we don't know much about its binding affinity. How do we design these systems? And so we were a little bit curious about that. And so the postdoc, Matt Copeland, made a series of different truncation tails where he kept the same N-terminus and then targeted longer and longer sections of this LAC operator. He also took two different promoters from the Anderson promoter library that's used for the iGen projects all the time, one that's very weak and one that's close to a maximum housekeeping promoter. And so you can see that even as you get really, really short in terms of the number of bases that these tails target, you can still get a very strong degree of repression. So that's the dark bars here. This is a protein production rate as a function of the tail length. And so you'll see that you have to get down to about 5.5 repeats before you see a strong GFP signal from these promoters. Instead, if you have a weak promoter, however, you'll see that the drop-off occurs at a much longer tail. And so we have a lot of space to play with. The other thing that we noted was, well, if we can drop it below 5.5 bases artificially, then we'll lose this DNA binding ability. And so what Matt decided to try was putting in different protease sites throughout the protein. And so originally we designed it to make 5.5 or 5 or 6 mers using the TEV protease. And so the TEV protease in E. coli has no natural target sites in the E. coli proteome. So you can express in E. coli, it has no problems. It's not going to alter gene expression. And so what he did then was he put this under the control of an inducible promoter and then targeted it to the particular TAL with these cleavage sites. And he did that by introducing in the loops that are later on when the crystal structure came out. These are solvent exposed, and so they're sticking out. And he just literally appended them in between the two repeat domains. And so when he put this system together into E. coli, the way that this works is you've got a reporter protein here, the TEV or its inactive mutant, and the tail, which either is the one without protease sites, or I'll show you the one with protease sites here in a minute. And so if you don't have the reporter protein, this is what it looks like. If you don't have any of the repressors, this is the maximum expression you're going to get from this particular system. If we have the normal tail, whether it's with a TEV or inactive TEV, you see we don't get much expression even when we induce the TEV protease. But if we take the system where we have the TEV protease sites encoded into the tail and we have the wild type TEV protease, it cleaves and chops up this particular tail, and you relieve repression, and you get basically the same level of expression. More interestingly was when we took the inactive tail, and so when it's this active site mutation renders TEV able to bind but not able to cleave protease target sites, we still saw some level of induction. So I'll get back to that in just a second. When we express all these systems, we see no growth defect, and if you actually look at it relative to LAC-I, it's more off in the off state and more on in the on, or almost as on in the on state. So it actually has a larger dynamic range than the well-studied LAC-I systems. And so this works as we expect. When you look for the protease, it's destroyed in the case where the active TEV is expressed. But in the inactive TEV protease site, you'll see that it's still hanging around. And so this is something we're really interested in because it means that just perhaps binding, this is the hypothesis, binding to the tail is preventing its ability for the tail to bind to DNA. And so we tested that with a second mutant in the TEV protease that basically increases its KM. And so this ability to turn on gene expression, so this is the normal tail TEV system, this is the inactive, so this is what you could get if you turned it on. This is what happens with the inactive TEV protease, and this is with the KM mutants, and you see that you don't have nearly the degree of expression. And so what we think is happening is that the TEV protease is just binding and sequestering away the tail away from the DNA. So that would have a lot of profound effects that we're going to explore here in the near future. So I think I've left about 5 minutes for questions. I'd like to leave you with the following take-home messages. Hopefully I've convinced you that biotechnology has a role to play in sustainable chemical production. Hopefully I haven't convinced you that this is a foolish PhD that you've ventured onto. But I do want to make sure that you know that it's not a silver bullet. It's not going to work for all chemicals. We're not going to be the form of the chemical industry in the future. I also hope I convinced you that synthetic biology is not new. It's just the latest evolution of what I think is a fascinating field, and chemical production is just one exciting application of these technologies. I think cyanobacteria have a part to play in this. What that role is going to be defined in the future, and I think in today's economic terms it doesn't look very good. But if all of a sudden you start thinking about a world in which there's a carbon tax or some sort of CO2-based economy, things may change. And I think these transacting regulators, whether it's the tails, the CRISPR-Cas systems, or the small RNAs, if you can do the same type of technologies, I think these are going to really enable even more advanced, more sophisticated ways of metabolic engineering for cells. So I'll close with the group again, of course, and I want to acknowledge a large set of funding sources, both in terms of fellowships that the graduate students work really hard to earn, as well as grants and other contracts. So I think I've left 5 minutes. I'd be happy to answer questions, and of course, please approach me up after lunch.
Learning for the fatty alcohol enzyme, can you compare the 4-electron reductase with the 2-electron reductase that you showed in our study? And also, that form fat batch fermentation for the titer, or
Yes. Yeah, so the titers that we would get in just a batch study are much lower than what we had in that fed batch approach. And so we think we can even improve it further if we had some sort of more sophisticated profile. The real question becomes, is that actually a practical production strategy? Are you going to be able to use biomass feedstocks and apply a fed batch strategy that would get you the same levels of titer? That's a great question I don't know the answer to. The first question was, we compared 3 different acyl-CoA reductase, acyl aldehyde reductase systems, and we found in our hands that the best activity came from the fused enzyme, which was from Marinobacter hydrocarbon classicus. It's a mouthful, but there are other groups who have compared others and seen very similar results.
The tail system you're talking about, do you plan on generalizing that to other systems?
Yes, I pulled that for time, but what we've ended up doing was it would have been a real pain in the neck to try to introduce those TEV sites throughout the tail as we have. And so what we've done is we've put them just at the front, the N-terminus and the C-terminus. It turns out you only need the N-terminal one, but if you have both, it enhances the dynamic range. And so what we've designed is a family of vectors with those 2 cleavage sites, and we can drop in a neat codon synthesized sequence for the DNA targeting part of the tail into this vector, and so it will then express. And we've done that for 3 different target promoters, and it works equally well in all 3 cases.
Another question about the tail. Maybe 2 questions. One, do you have to look for different regions in that lac eye in order to get a strong repression, like with the dCas9?
Yep.
You also have to make different guide RNA to get the right position. And then the second question, have you compared it to dCas9?
Yeah.
What the relative efficiencies are?
So the first question is a great one. So there is literature that shows that there's an absolute requirement of a T in the first position of the tail binding target. That's not true. So you can remove it, you just need more tail. So it enhances the binding affinity. I don't know the numbers because nobody's measured them. But we found that if you mutate that base, you can still get affinity, but you just need the stronger promoter on the tail in order to get the same degree of repression. So we think we can then have flexibility to perhaps tune the degree of repression. And so kind of taking your question and flipping it instead of it being a challenge, we could actually use it as a tool. But we don't know the rules yet. And so that's one of the things that we're very interested in studying, trying to figure out exactly, is there a nearest neighbor code for 21 target sequence or target bases is a happy medium, or we don't know those rules. There's some evidence from the TALEN literature that it actually doesn't matter about the length, it's the number of G's or the number, I forget which base, but one of the bases is more pronounced in creating the effect. And there's a lot to be learned here, but because CRISPR-Cas has exploded onto the scene, it's left a really nice research hole for my group because tails aren't being so. We actually think because of the specificity, in these particular applications, they may be superior to the CRISPR-Cas system for that regard, but you've got to make these huge proteins every time, and so that's a challenge. And so it may be better in the long run, and we put this out in a current opinion piece earlier this year, that for exploratory purposes, the CRISPR-Cas system may be the way to go, because you can synthesize lots of different guard RNAs fairly easily, but then you would probably implement it permanently with a tail. Now, the second question is the subject of one of my students' current proposals. And so, yes, we would love to know from many perspectives, binding affinity, off-targets binding, energetic costs. So in the case of small RNAs, right, these are things that are turning over constantly, and you're making the messenger RNA constantly. So that's a lot more energy. So is that a more expensive way of implementing metabolic engineering strategy than just having a single protein, or in the case of the CRISPR-Cas, a single protein with a guide RNA? And so we're looking at that using metabolic modeling to try to project energy costs, and we're also trying to design a single reporter system where we can target with the same affinity or the same degree of repression all three systems, and so that we can then do a comparative experiment. So in 2 or 3 years, I'll tell you the answer to that one.
So, really interesting talk about the counter selection system. I'm curious just to know how generalizable that is for other kind of cyanobacteria, and also, a lot of counter selection systems have high escape rates.
Yep.
And I'm just curious how this compares to other canonical counter selection systems.
So we tried a whole range of counter selection systems, SACB, 5-fluorouracil, and I forget the others. And none of those worked in our hands very well. This is not perfect. You do have the occurrence of spontaneous mutants, and it is sensitive to both light intensity, temperature, pH, so there are optimization steps that we've since figured out, and I couldn't repeat to you. We'll have to send it to you. And so it works really well. We've also tested it, I believe, in 6803, so that's a freshwater species that people use a lot. We never got around to testing it in 7942, which is the other common species that people works with. But I believe they all have the same acyl-CoA synthetase homolog. It's just a matter of whether there's another one someplace else, and we haven't done that full search.